Before we take another look at the steps in DNA replication, let's take a look at what are the components in the DNA structure that explain why we need the steps that we need in DNA replication. So let's look at some rules for DNA replication. The first rule is that all polymerases, this includes DNA polymerases, RNA polymerases, things that you might not realize are polymerases because they have different names, but still are polymerases because they synthesize DNA or RNA, such as primase, which synthesizes the RNA primer, is an RNA polymerase, or telomerase, which is a type of DNA polymerase. All polymerases, DNA polymerases and RNA polymerases, can only add nucleotides to the 3' end of an existing nucleotide. So that means that the new growing strand has to grow from the 5' end to the 3' end. So the new nucleotides are being added to the 3' end of the growing strand. The numbers are given because of the 5-carbon sugar that forms DNA and RNA nucleotides. Here in the top we have a DNA nucleotide, and the bottom an RNA nucleotide. On the 3rd carbon in that sugar, both have an OH, so that is the 3' end, while on the 5th carbon of that sugar, you have a phosphate attached to it, so that is the 5' end. And the new nucleotides are attached to the OH in the 3' end. This is all polymerases can only attach nucleotides on the OH on the 3' end of the growing strand. So this means that since the DNA is anti-parallel, one strand is facing in one direction and the other facing in the other direction, the complementary strands will be synthesized in opposite directions as well. The leading strand is synthesized in the same direction as helicase is opening up the DNA helix. Leading strand is synthesized from the 5' end to the 3' end, and for this strand, that direction runs in the same direction as helicase is opening up the DNA helix. Every time you put a nucleotide here, you will have a 3' end available, so you can add another nucleotide. But for the lagging strand, since the direction of the lagging strand is in the opposite side, the direction in which that strand will be synthesized, which is the 5' to 3', is in the opposite direction as helicase will be opening up the DNA helix. The other requirement when you're synthesizing either DNA or RNA is that you need a template, because the template is what tells you what order of nucleotides you will be adding. If you don't have a template, you don't know which nucleotide to put on, so you cannot add nucleotides. So you need to have a template, and then you add nucleotides matching the existing bases that are present in that template, and that gives you the sequence of nucleotides that will be added. So this is true for both DNA and RNA. You need to have a template so that you can synthesize a complementary strand that is complementary to that template. This third rule is only for DNA, and DNA nucleotides cannot be just added to a template. They need something already there that they get attached to. So if you put a DNA nucleotide by itself on a template, it's going to fall off. It's just not going to stay. The reason for that is because of the difference in structures between DNA and RNA nucleotides. DNA has the name for deoxyribose. Ribose is the sugar that forms part of DNA and RNA. RNA just have ribose, and DNA has deoxyribose. Deoxy meaning is missing an oxygen. So if you look at this position here, RNA has an OH, but DNA is missing that oxygen. If you remember from our previous videos, oxygen is a very electronegative molecule, and molecules that have a lot of oxygen are more polar because oxygen makes this uneven distribution of the electrons, pulling electrons towards it. So that makes even a neutral molecule like water is polar because the oxygens are pulling the electrons towards it. So a molecule that has more oxygen will be more polar. That means RNA will be slightly more polar than DNA, and if you see the environment in which the DNA replication is happening inside the nucleus of the cell, it is a water-based environment, it's a polar environment. So RNA will be slightly more stable in that environment than DNA. So that's why DNA nucleotides do not stay there by themselves. But RNA nucleotides can. So the way to solve this is that before you put DNA nucleotides, you have to put RNA nucleotides. And you put a couple of RNA nucleotides first, since they don't need to be attached to anything prior. And once you have a set of RNA nucleotides attached to your template, then you can start attaching DNA nucleotides. And notice that every time you're attaching a new nucleotide, you are adding it to the OH on the 3' end of the growing strand. So that's what we'll be attaching the new nucleotides. Every time you put a new nucleotide, you attach it to the OH of the previous nucleotide. So this segment that we have here of RNA, this is what we call an RNA primer. So that's why you need to start with RNA before you can start putting DNA nucleotides. That's why you need a primer. In this example, we're using 3 nucleotides as a primer, but in reality, primers are somewhere between 10 and 20 nucleotides of RNA that are put first before you can start putting DNA nucleotides.